Morning, everyone. Welcome to session 16. My name's Ed Topp, and I'm going to have the honor of introducing Professor Lance Price this morning. So, Lance Price is a professor at the Milken Institute School of Public Health at George Washington University. He works at the interface between laboratory research and public policy to address the crisis that we're all familiar with, antibiotic resistance. He and his team are using genomics to pioneer new strategies for source attribution and trying to develop new strategies to stop the spread of these microorganisms. His research on the connection between AMU in industrial agriculture and drug-resistant infections in people have been widely published and mediatized in the newspapers. Prior to joining the university, Dr. Price directed the Center for Food Microbiology and Environmental Health at the Translational Genomics Research Institute, and he received his PhD at Johns Hopkins Bloomberg School of Public Health, where he was a Livable Future Fellow, apparently. And Lance, it's up to you now.
Thanks, Ed. Well, it's a real honor to be here. I've been super impressed by the talks and all the presentations, and I love Quebec. I mean Montreal. Sorry, I love Quebec too, actually. And it's sort of like the U.S. but civilized. It's really pretty neat and clean. I live in Baltimore, so everything looks a little better. But, so today I want to talk about our efforts to quantify the global impact and inequities associated with food animal E. coli. And so just to, I'm going to talk a lot about how we're tracking E. coli and less about the resistance. I'll touch on the resistance, but I feel like that's central to getting at the resistance question, so I hope you're not disappointed. So really, this is rooted in my long-term interest in understanding or quantifying the contribution of antimicrobial use in food animal production to antimicrobial resistant infections in humans. I've recently sort of opened this up a bit. I'm also now very interested in antimicrobial use in plant agriculture. If anybody saw those two great talks on antifungal resistance a couple days ago, then you'll know this is also a really important topic. And so we know we've talked over the past few days about how this these can be connected, and one is through horizontal gene transfer. So you use antimicrobials in food animals, you drive the evolution of new antimicrobial resistance, mobile resistance elements that can then be transferred into human adapted pathogens and increase the burden. You can also have problems with direct infection. So using antibiotics in animals, driving the evolution of zoonotic pathogens, resistant zoonotic pathogens that can then directly cause drug-resistant zoonotic infections in humans. And this is really the area that I'm most interested in. Well, I'm interested across the board. This is the area that I feel capable of working in. Horizontal gene transfer is tough. So I've worked on Staph aureus quite a bit. So, a lot of the work, early work was done in Salmonella and Campylobacter, the kind of classic frank foodborne pathogens. But when I was looking in meat and animals, I was seeing all of these colonizing opportunistic pathogens. They're the biggest, the world's biggest killers, and they seem to have been, people were ignoring them 20 years ago. And so, I started working on Staph and E. coli, and E. coli is really my favorite bug today. And it really deserves our attention. So here's just 4 figures to hopefully underscore that. E. coli kills 1 million people, roughly 1 million people around the world each year. It's ranked number 1 in terms of bacteria that are directly attributable to or associated with fatal antimicrobial resistant infections. I think, importantly, a lot of people, at least in the lay public, when they hear E. coli, they think diarrhea. But they should be thinking of UTIs and bloodstream infections, because 94% of the infections that are killing people around the world are not diarrhegenic. They are extraintestinal infections. And then this is a social justice or environmental justice issue. 4 people in low and middle income countries die, four times as likely to die of E. coli infections than people in high income countries. So 4-to-1 rate. And so this is really, again, this is an organism that deserves our attention. But despite its dominance as a global pathogen, we really know little about its its ecology. We know its name. And I really do believe that understanding the ecology is critical to developing, effective intervention strategies. So one thing that we do know is it's an incredibly versatile organism. So it can colonize. It colonizes the guts of all the major vertebrate animal species. This is not a very globally representative image here. I should have ducks and geese and sheep and goats as well. But it also colonizes our really cool clip art human being. This is our avatar for humans right now, according to Microsoft. And so, it's very broad host range. And it also has this broad range of virulence. So they all have the same name, but they range in virulence from commensal, benign, living in your gut. Most of you are sitting on it now. To dirogenic, the kind that cause these dramatic foodborne outbreaks, and then the kind that cause urinary tract infections. And as I mentioned before, mortality really skews towards the extraintestinal pathogenic strains. So XPEC is a term coined by Jim Johnson formerly at University of Minnesota. Jim recently retired and is now dedicated to playing bagpipes. But he still consults with me. I love Jim Johnson. So XPEC, they have these special virulence factors that allow them to cause disease outside of the gastrointestinal tract. Like, think about fimbriae that can lock down on urethral cells, even under sheer force. And so as you're urinating, they actually lock on tighter. They can cloak themselves from the immune system. They can acquire iron. They can do all these great things, bad things that allow them to be good pathogens in the urinary tract. And so they cause, in the US, 6 to 8 million bladder infections, 250,000 kidney infections. I think that's an underestimate. And then 36,000 to 40,000 deaths in the US. And antimicrobials are critical therapies. So people, talk about cranberry juice, but I would use the cranberry juice to take an antibiotic if you have a UTI. So, and I think when you think about mortality too, you've got to recognize the fact that the urinary tract is the gateway to the blood, is a major gateway to the blood. So more than 50% of urinary tract infections start with the urinary tract. And then, again, Jim taught me this, 85% of urinary tract infections are caused by E. coli. It is the #1 pathogen. And these strains live in our guts, the XPEC strains live in our guts without any symptoms. And then they cause infections when they make that short trip from the anus to the urethra. Most XPEC infections are sporadic. So this is a really important point, and one that I'll touch on a lot in this talk, because it dictates the kind of tools that we use to understand spread and source. And so we've been asking this question, well, how do we get these XPEC in our guts? And so we know that these are more, another avatar from Microsoft here. We know that human-to-human transmission is really important. And whether there's an intimate relationship here, we don't know, but we do know that the world's covered in a thin layer of poo, and we're sharing E. coli all the time. And so who we live with is a big risk factor. But we also think that this is important, right? So that people can get exposed to the E. coli that are in the guts of animals. Now, whether they take up residence and cause infections is really important to figure out. And we think that meat is an important vehicle. So there's direct exposure to the animals, so people living around or working with these animals, and then, particularly in high-income countries, most people are probably exposed through meat handling or consumption. So I first started studying E. coli when I was working at the Translational Genomics Research Institute in Flagstaff, Arizona. And this is a small mountain town in the U.S., 70,000 people about. I went to college here, actually, way before I was doing this research. And it turns out that this is was a great place to kind of start these investigations, because it's because it's geographically isolated and there's no food animal production, we could really look at meat as a vehicle and really try to understand what's coming in and whether those bacteria are making their way into people. And so we set up a study here where we simultaneously collected E. coli from urinary tract infections and blood infections, bloodstream infections from the only hospital in the city. And my classmate just happened to be the head of infection control there, and so she helped organize that. And then there's only a handful of grocery store chains in the city, and so we went to every grocery store in the city twice per month and sampled chicken, turkey, and pork, every brand. And we brought it back, and by the end of the year, we'd sampled, well, by the time we were done with the genome sequencing, we'd sequenced almost 2,000 meat isolates and 1,200 from people. And at the time, this was a while ago, more than 10 years ago now, this was sort of an unprecedented study, and I was really confident that we were going to nail this. We're going to use genomics, we're going to sequence the genomes, we're going to draw trees, and we're going to figure this out, right? And so, the question was, what's the population overlap? And so initially we started with this three-step process. We were going to sequence the genomes, extract an MLST signal from those genome sequences, and then we were going to cluster them by sequence type and then do core genome phylogenetics. Now, we'd been using core genome phylogenetics to study the evolutionary history of things like Staph aureus ST398, to study the origins of the Haitian cholera outbreak following the tsunami there, or sorry, the earthquake there. And we'd been using it to study the evolution of sequence type 131. And so I was, again, very confident that we'd be able to do this. So the first thing we found was, hey, there's a lot of sequence types, so there were more than nearly 450 different sequence types among our collection. Each of the bubbles with a number in it, each number represents a sequence type. The color of the bubble, size of the bubble represents how many isolates had that sequence type, and the color represents whether it came from people or meat. So red for meat, yellow for urine or people, right? And so what we saw was that certain sequence types were disproportionately associated with meat, others were disproportionately associated with humans, but you saw these mixed pies, right? So we decided to start with ST131 because we had so much experience working with this organism, and all the previous studies suggested there was no meat involvement, no food and animal involvement in this pandemic strain, yet we had this big red pie piece. So we sequenced the genome, we took the core genomes and we got rid of the recombinant regions and looked at the SNPs. And sure enough, we saw the pandemic lineage, and there were no meat isolates clustered with those pandemic strains. So this sort of held up with what we'd seen before. But we also had this other cluster of the FIMH22 lineage. Now, these are also causing infections, including bloodstream infections, and they're clustered with meat. So to get a higher resolution tree, we separated these H22 strains, and we drew another core genome phylogeny, this time rooted. And what we saw were these two clusters of where we had tight clusters of human isolates and meat isolates. Now, no clonal relationships, but we saw this clustering. So we thought we nailed it. We wrote the paper up and we sent it out for review, and the reviewers came back and said, basically, you didn't nail it. All you've shown us is that these are related, and in fact, some of your human isolates are basal to your meat isolates. How do you know that people aren't contaminating the meat? And then I imagined people peeing on chicken in the store, and and so I have an active imagination. So but what this did underscore for us was that we were struggling to cope with this enormous underlying diversity of the food animal E. coli populations. And so, you just think about a single poultry house, right? So they can have 100,000 birds in a single house. That's pretty typical, 50 to 100,000 birds. How many samples would you want if you were to define the E. coli populations that were colonizing all those birds? Probably want a few, right? Now, this is a map of poultry production in the United States. Each of these blue dots is 1 million broiler chickens each year. So we produce billions, 9 billion roughly, chickens each year in the United States, mostly in the broiler belt, sort of overlaps the Bible Belt, interestingly, and then some in chicken there. And so now how many samples do you want? it is impossible to sample these populations sufficiently to define the E. coli populations. And what I realized is that we were using these tools best suited for outbreak investigations. So outbreaks, you have a point source, you have a young clone emanating from a single point source, and you can trace those back based on time and epidemic curves, but also using core genome phylogenetics, where your epidemic clones, your outbreak clones, would be clustered with few or no SNPs. You can count SNPs and say it's part of this outbreak. But in the case of sporadic spillover from a massive food animal population, where people getting exposed does not necessarily translate to immediate infection, it just transient gut colonization. And sometimes those will progress to an infection. So these are truly sporadic infections, but a massive, probably continuous spillover. And so if you draw a tree from these, this is just a fake tree, you're not going to see any clonal relationships. And we saw that in the real tree, the ST131 tree. So even if you isolate a single sequence type, you're not going to see many clonal relationships because it's, again, impossible to sample enough. But, stepping back then, I had plenty of time to step back because this was like 10 years of beating my head against the wall and a couple of depression cycles. And, and this was a hard period for me, right? But stepping back, I realized that, the important thing that we were trying to evaluate was not which farm did it come from, because we can't even get to a farm in the United States. They've, successfully blocked that. It's not a particular slaughterhouse or an animal. It's, did it come from an animal host or a human host? That's the first question that we want to answer. And again, let's dig a little more into how these epidemiologic investigations are performed, right? So we're using, SNPs, or single nucleotide polymorphisms from the core genome to, infer phylogenetic relationships. But it turns out that the accessory genome is where you find the genes related to niche adaptation. So, mobile genetic elements and other accessory genes that might come in slower, through recombination, could lead a strain to become better adapted to a human host or animal hosts. And so our hypothesis here is that if you have a spillover from, say, a chicken host into a human host, immediately after that spillover, a recent spillover, you can just look at those host-associated mobile genetic elements and infer that came from an animal. Over time, as if it gets established in the human host, it's going to adapt to that host, and it's going to change over and pull in mobile genetic elements specific to the human host. And so at that point, we wouldn't recognize it based on the mobile genetic elements, but it doesn't matter anymore because it's not actionable in the animals, right? It's not actionable in the food supply anymore. It's actionable in people. That's a different challenge. And so this kind of reminded me of Forrest Gump, sitting on the park bench. I don't know if hopefully you've seen Forrest Gump, but he's sitting on the park bench and he sees this woman and he says, those look like comfortable shoes. This is my bad Forrest Gump imitation. Mama said you can tell a lot about a person by their shoes, where they've been, where they're going. And I thought about these mobile genetic elements in these shoes, right? I know she's a nurse just by looking at what she's wearing. And I want something that easy when it comes to E. coli. So we started looking for mobile genetic elements, or the shoes that E. coli wears when they're in chickens or pigs or cattle. And so the first one that we looked at was coli plasmids, and that's because we work with the guy, not Jim Johnson, but Tim Johnson, they're collaborators, in Minnesota. And he's had years of working with coli plasmids, and he said, let's look for coli plasmids, because I know these are associated with poultry. And so we looked in our poultry isolates, and 64% of the poultry isolates were carrying the coli plasmids, and 5% of the humans. So these are really, I think these are the shoes that E. coli wear when they're working in chickens, right? And so, we decided we'd use this marker. And so, now if you have a person with a UTI, we get a culture from her, and then we isolate E. coli with these chicken shoes, then we suspect that this probably originated from a poultry exposure. So we went back to our tree now, and now we have these two clusters that we're interested in, and we overlaid the tree with the coli plasmids. Look at this. I mean, this very much changes our perspective on the direction and the recency of these transmissions. Right, so we didn't sample the population enough to recognize any clonal relationships, but the human isolates are carrying these coli plasmids that are clearly for poultry adaptation and virulence. And so we went back to the genomes, and we knew that E. coli had this massive accessory genome, and we started looking for more of these mobile genetic elements. And we found 11 more associated with one or more food animal species. And I can tell you we overfiltered, so we're going back. I'll talk about that later. And then we found 6 associated with humans. Now, we then struggled a little bit with, like, how do we merge these together? And so my colleague Dan Park and Jinka Wu and their colleague Tom Bays, they suggested that we use this Bayesian latent class model. And so we can combine these and get the benefits of increased sensitivity without taking a total hit on specificity. And so the way this works is, you get a person with a UTI. You get a culture from a person. You get the E. coli. You culture the E. coli from there, and you want to know whether it came from a human or food animal. So initially, you have this, prior probability of, the distribution of strains and the probability of a food animal or human host. But you can inform that probability then by running MLST, maybe a sequence type. I'm not saying 69 definitely is associated with food animals, but let's say that modifies your posterior probability. And then you start running start analyzing your host elements, and you get this different posterior probability. Anyway, so by running this model, we can actually have a quantitative probability that a sample came from a particular host. So we can, and we've set this, semi-arbitrarily set this cutoff of 0.8. So if it's an 80% or better probability of coming from a particular host, we're going to say that was the host. And so now we have this model that we can apply, and we applied it then to our whole Flagstaff collection, and we found that 8% of the UTI isolates had an 80% or greater probability from being from meat. When we applied the same model to the meat isolates, we found only 0.5% of those isolates appeared to be of human origin. And so this told us, hey, we're probably handling our samples pretty well, and the direction is mostly from food animals to people. Now, 8% may sound, we call that foodborne zoonotic E. coli, and we published a paper in One Health on that. Now, 8% might sound like a small number, but if you scale that to the United States, we're talking about more than 500,000 infections a year that are UTIs that are of zoonotic origin. But Flagstaff is a young, homogeneous population. They're, socioeconomically and ethnically pretty homogeneous. And so we wanted to know if the rates were the same in more diverse states. And so we, as part of another study, we were collecting lots of E. coli isolates from, we collected lots of E. coli from meat and humans in Southern California. And this is a collaboration with Sarah Tartoff from Kaiser Permanente. And by the end of the study period, we collected over 3,000 E. coli from meat and sequenced the genomes from meat, and over 2,000 from clinical. These were just UTIs. And then we applied the model to those as well. And what we found in California, Southern California, was that 18% of these had a 80% or better probability, and most of these were actually 90% or 90% probability. And so, on the scale of 1 in 5 human UTIs in Southern California appear to be caused by E. coli from food animals. Now, if you look at the sequence types, this shouldn't be very surprising if you know anything about phylogroups and extraintestinal virulence. But basically what we know is that, that extraintestinal virulence is associated with phylogroups B2 and D. And so the E. coli, the clinical E. coli, were disproportionately of the B2 group. those that weren't called FSECs. The ones from meat were disproportionately A and B1, which is not a big surprise. I mean, that's what we would anticipate. But the foodborne zoonotic strains appeared to be disproportionately B2. It was a mix of the two sort of populations. And then when we plotted the top 1% of the strains that had 1% or more of the FSEC infections, the sequence types there, and then we plotted those that were in 1% or more of meat on this graph. And what you see is that you can think about prevalence in meat as being stochastic potential for spillover. So the more of it in the meat, the more likely it is that somebody's going to come in contact with it, right? So just think about that as not inherently, nothing to do with inherent virulence, but just opportunity, right? And then those that are sort of going to the right on the x-axis, that probably has more to do with inherent virulence. And so, these that are sort of lining up on the, 0% of FSEC infections, but fairly prevalent in meat, we'd say have very, little zoonotic potential. But those that are fairly low in meat, but causing a lot of FSEC infections, we would say probably have inherent virulence. And there's no surprise that we're seeing, again, mostly B2 and Ds here, and mostly the A and B1s in the previous box there. And then we have these oddballs in the middle that may have some special virulence. But we're saying that perhaps all of those at 1% or better of the FSEC infections are potentially of concern, and so we want to dig into those deeper. And one other point about these. When I talk about these specific strains, some of these strains with my colleague Tim Johnson at University of Minnesota, he says, hey, these are the same strains that are infecting the birds, right? So this is a potential win-win between public health and food animal production. We're often at odds with one another, but in this case, if we could eliminate these or reduce these in the animals, we could have a public health win and a food animal production win. So this, though, seeing this difference, nearly, more than twice as frequent in Southern California as in Flagstaff, made us, ask, why. And so we started looking at different factors, and we found that that the FSEC infections really clustered in the poorest communities in California. And so this was very intriguing, and in fact, there was a dose-response relationship. So with each 5% increase in poverty rate, we saw a 14% increase in zoonotic transmission. So why is this, though? So now we see that there's this correlation with poverty. Why? And, so it could be behavioral, it could be structural, so maybe, less opportunity for good hygiene. It could also be the quality of the product. So I'll just, since I have a little time, I'll tell you, when we've done studies in Washington, D.C., and we went to the different wards to buy poultry products, what I was struck by when we went to the poorest wards and went to the poorest grocery stores, that these packages of meat would be bloated with saline. So they're ripping these people off, I mean, they're bloated with saline. When I put it on the belt in the grocery store, stuff would be dripping out onto the conveyor belt, where people also put their vegetables and their candy bars and everything else. And I think people in the poorest communities might be paying the same price for the actual meat as people who are buying organic that has no saline at all in there, right? And they're just paying for the meat, because water's pretty heavy. Right, and so I think that these are factors that we have to dig into. That's only anecdotal, but I mean, I think that these are possible ways that these are spreading. But now let's think globally, right? So that's just in California, right? Again, in a high-income country, but with quite a diverse population. So these infections could vary greatly with cultural practices, with water sanitation and hygiene, and animal production. Particularly in low and middle income countries, right? And so we're really interested in looking at whether zoonotic E. coli infections are more common in low and middle income countries than in high income countries. And specifically we're interested in bloodstream infections because these are the ones that kill, right? And so we have been scraping the public databases. And so Interobase is one of our favorite places to go. So we go to Interobase and we get all the E. coli genomes and we sort them for, we identify those with bloodstream infections, UTIs. We're also including UTIs now, urinary tract infections. We're getting all the meat and animal isolates. And we're analyzing those genomes using our models. Now, our models were adopted or developed on American isolates. So we clearly need to make sure that it's trained properly for these global isolates, but I think this is an important view into what might be out there. But there's one thing I have to point out that I think is a travesty. So we're getting samples from high and low and middle income countries, but 94% of the E. coli genomes are coming from high income countries. I already said that the E. coli mortality, people in low and middle income countries, there's a 4-to-1 ratio of people dying in low and middle income countries to high income countries. So this is a data gap that really has to be filled. And we have to bring money from the high income countries to do this. And so this will be one of my career goals for the next few years. But when we applied the model, again with the caveats that I pointed out before, we saw that the high-income countries across the world were pretty consistent with what we saw in California. So about 17% of E. coli isolates, bloodstream isolates from high-income countries appeared to be zoonotic. But in low and middle-income countries, it's closer to 29%, which really is consistent with our hypothesis that a breakdown or a non-existent water sanitation hygiene, more intensive human-animal interface is probably going to lead to more of these transmissions. So think about this. 1 in 3 human bloodstream infections in low and middle-income countries could be caused by E. coli from food-animal production. And so I'm going to just flash a comment up, but I have to caveat. When you go to public databases and you scrape those genomes, you don't know the bias that went into sampling those. Were they looking for drug-resistant strains? Were they not? How are they sampling? This is a hypothesis-building exercise so that we can go out and purposely collect samples in an unbiased fashion. But we found that the strains, the E. coli, the FSEC strains from low and middle income countries were 3 times as likely to be resistant to 10 or more classes of antibiotics than those from high income countries. So I think, again, we have to take it with a grain of salt because of the potential bias, but it definitely raises an eyebrow for me. And I'm very eager to get out and sample in an unbiased fashion. But if you just scale from these percentages and you scale with the number of people living in and the number of people dying of E. coli and then the percentage of populations living in low and middle income countries and high income countries, that would translate to about 250,000 zoonotic E. coli deaths per year currently unattributed to food animal production. I think that this is really important, and think about the ramifications for policy and how we prioritize the money that's going for antibiotic interventions and just interventions in general. So our next steps are to identify more host-specific MGEs. We over-filtered, and we know this because we've gone back to the genomes, and we see that when we were trying to find really universally informative mobile genetic elements, we are missing a lot that might be specifically informative for different species. And in a Bayesian model, you can actually, you can use those without too many hits. So we were finding, these types of elements that were sort of, consistent with more than one animal, but we could differentiate from humans. So now we're looking for these that are specific to specific animal hosts. And then we'll revise the Bayesian latent class model to predict specific hosts. I think this is critical to then developing interventions that are specific to the different animal hosts. If you want to go out and vaccinate or develop probiotics or phage, you can apply it to the right animals. So we're expanding our hosts to include regionally important species like ducks in Asia, right? So if you look at total animal production, it may not be that important, but in some regions, people might be getting exposed to a lot of duck-associated E. coli. And so we need to consider those regionally important organisms. We want to make this tool, after we do this next series of refinements, we're working with David Annenson from Pathogen Watch, and we'd like to get an instance of this up so that people can just upload a single genome. You don't have to do a massive collection to understand whether your E. coli potentially came from an animal or not. You can just upload one genome and we'll put it in context and give you that score. And then we want to quantify transmission of antimicrobial resistant strains between food animals and humans. So again, it's so critical to sort of dialing in our interventions. And finally, identify the highest risk strains and their reservoirs globally. So which strains are the most important in West Africa, which ones are most important in the United States, and then develop. Our long-term vision is develop strategies that can decolonize people and animals from the highest risk strains with a combination of probiotics, phage, and vaccines. But before we do any of that, we've got to close this digital divide. Remember the digital divide with the internet and libraries, and we had to bring everybody up to having the same access. We've really got to close this knowledge divide here and get something closer to 50-50 here. And so with that, I just want to thank all my colleagues, especially my team at George Washington University. We make them work really, really hard. And I have this great, my colleague and wife, Cindy Lou, she's the only reason any of this works, because I'm a flake. And Yashan Wang, who's our PhD student's amazing. Maliha Aziz, Dan Park, Ed Sung. And Vanessa, and then Mark Steger from Staten Serum Institute. We're working together to try to close this knowledge gap or this data gap. And who else? The funders, of course. And again, the organizers. Thank you so much.
Wonderful talk,
Thank
Lance.
you.
I'm really impressed by this excellent work. I have a question about, so you've sequenced lots and lots of clinical strains, lots of animal strains. I didn't see any data on poo strains from humans.
Yeah.
And I just would assume that there could be differences between the strains that cause infections in us and the strains that usually don't do that. And in particular, in terms of if we're trying to figure out, does the bacteria on a piece of food come from humans? I would assume it's more likely it comes from poo than from a bloodstream infection.
Right.
So have you done a lot on poo strains as well, and what's your thinking about that value of that?
My thinking is that you're exactly right. I mean, we're definitely, our model is biased towards the extraintestinal strains. And so, some of those mobile-genetic elements actually might be related to virulence. And so I think I know we're trying to close that gap. And so the more, we're working with Evgeny Sogyurenko. We're trying to get some of his colonization strains, and we're looking for globally representative ones. So it's clearly a gap.
So my question is, do you think that the model that is emerging is that humans have a commensal E. coli that is primarily ST131, and then you have these transient E. colis that are coming perhaps from food that are bringing these mobile genetic elements to the commensal flora that is primarily ST131?
Well, so I think you're right that ST131 is a really important, it's a very sticky strain. So I did some work with Jim Johnson, and we were enrolling cohorts, or people at the VA, and then their family members, and we were expecting some turnover. We were going to see what the which microbiome organisms were associated with loss of strains, but they just never lost them. Once they got them, they were just holding on to them, and our cohort just started going like this, right? And so I do think that people are co-colonized. I don't think we have a lot of great tools for doing that. I mean, one of the things that we're trying to develop is something called an ecolome set of primers that will allow us to go into a single sample and define the E. coli diversity within a mixed diversity within a single sample. I just don't think we have a lot of data there yet, but I do think that so going to I think you are also asking about horizontal gene transfer, right? And so, while I'm using mobile genetic elements to understand host attribution, there's also clearly mobile genetic elements associated with resistance that we have to consider. And I do think in some cases what you have is a transient colonization from a food animal adapted strain that doesn't do well. It gets stressed out in the human host, and it's going to be pitched out. That's one of the slides that I lost. But what they do is when they pitch out, some of those are going to be passed to other strains, human adapted strains, and so even at 131 might be able to pick up more resistance from a food animal adapted strain. So I hope that covered, yeah.
Hi there. I'm Justine. I'm an epidemiologist from the Yukon Territory up north here in Canada. I was just wondering, from a surveillance perspective, you said that you looked at the brands of the meats. Was the location of the manufacturing also noted? Just because regionally we can see the effects, but was there like transport from different areas?
So there's something called a P code that allows us to identify at least the processor that handled the sample. And so we're looking at how P code relates to resistance and contamination. And that's really important for the California study because we're trying to understand for a different study how resistance changes over time in California, specifically related to a policy called SB27 and outside of California. So we do have the data sort of, but we haven't done a ton of analysis with those.
Okay, very cool.
Thank you. Hi.
Very interesting talk.
Thank you.
Cornelia Smalla from JKI Braunschweig. You were studying E. coli from animals and from humans, but maybe the plants should be considered as well, as we heard yesterday from David, very understudied, but you can isolate from plants E. coli. They are not very abundant, but they can proliferate, and so I think they need to be in the picture as well. And actually, with respect to the plasmids, I think they can certainly be shared between the E. coli from the different environments. So I'm not sure whether they are ideal markers for the origin of an E. coli.
Well, so I agree with you about the plant piece. We definitely need to do more on that. I mean, because 9 billion animals all pooping, and what do they do with the poop? They use it as fertilizer, right? So spraying liquid pig waste on crops and then dry poultry waste, and we've done studies in the past looking at the survival of drug-resistant bacteria in those poultry compost piles. They're not actually composting them, and they survive way past the date where they said they're safe to apply to even edible crops. Yeah.
Great presentation.
Thank you.
I'm Pankaj Gaukar, PhD student from Auburn University, Alabama.
Hey.
So I had a question regarding the retail meat. Like when you are investigating the retail meat, are you able to get any information regarding was it antibiotic free or like antibiotic use? And if you're able to get the information, how accurate those labelings are?
Yeah, that's a great question. So I think we published a study about cheating. We had a paper in Science Policy last year about cheating in the beef industry. So I don't know if it's perfect, but I think it's raised without antibiotics. I think it's probably true for the specific animals. The problem is that they're raising them in the same houses. So the same companies that raise without antibiotics are also raising with antibiotics. They're using the same facilities. They're being slaughtered in the same facilities. Despite that, we do see a significant difference in the prevalence of drug-resistant bacteria on products with the different claims. But I think it's muddied from the co-use of equipment and barns.
Thank you.
Thank you for a good question.
Hi,
Steve.
Lance. Stephen Georgievic here from the University of Technology, Sydney. Really great talk.
Thank you.
So we've also been looking at the segregation of different plasmid lineages associated with different hosts. And we've found like coli plasmids sort of go everywhere. They're sort of quite host promiscuous, whereas there are some groups of plasmids that never circulate ever into animal production systems at all, that are prevalent in key top 5 or 6 pandemic expat lineages. So would you be able to speculate how you think, why a plasmid would never be found I mean, we screen 30,000, 40,000 genome sequences out of public databases. We never find them in animal production systems. So it must be a metabolic thing, or would you care to speculate on why that might be?
I'm not a very good speculator. Yeah, I don't know. I mean, I don't really have any deep thoughts on that. It's pretty interesting, though. I mean, I think that those are good markers, though. I mean, if they're solidly associated with one.
Oh, they're great. They show excellent transmission between companion animals and humans, and you find them in wastewater, but you certainly never find them in animal production systems.
So interesting. Yeah, I'll look forward to reading that paper.
Oh, yeah, thank you. Thomas Berendonk, TU Dresden. I really liked your talk, but I even more liked your probabilistic approach. But I didn't quite understand the slide where you showed the different isolates and the more XPEC isolates, and you said that if you have a higher number of isolates but a lower virulence, and then you showed
Yeah.
And those were then more zoonotic. I didn't understand this. And
Yeah, I didn't do a very good job on that slide,
no,
did I?
and I wanted to know.
No, I recognize that. Let's try again.
But my key question was actually, why do you put the threshold where you put it?
Oh, so that was arbitrary. I'm trying
okay.
to. Yeah, I should have said we need to decide what the threshold's going to be to decide which are the highest risks that we're gonna dig into farther. You know, because we need to. So let me do that, and then I'll come back and try to explain a little more. So if we can identify the highest risk sequence types, there's still more work to do because we need to understand whether zoonoses is a feature of the entire sequence type or if it's certain clades. So what we see with the ST131 is a perfect example, right? So there's the H30 lineage that's a human-to-human transmission pandemic strain. H22 looks like it could be, zoonotic, right? And so I think that that's going to be true with other lineages. ST117 appears to be it's an animal strain, right? And when you see it in people, it's almost clearly zoonotic infection. So what I was trying to get across there was that by plotting the prevalence in meat, I couldn't plot all of them because it would be a very messy slide. So we just took the 1% that were those that were in 1% or more of meat samples and 1% or more of the human infections, right? And then I plotted along the x-axis was prevalence among infections for that sequence type, and the y-axis was prevalence in meat. And so among the meat isolates. And so what I consider there along the y-axis is, again, kind of stochastic potential. If there's a bunch of it in the food supply, then there's a lot of opportunity to spill over. So if it was just about stochastic potential, then you would expect the higher it's on the graph to be the further on the right. But that's not what we see. What we see are a bunch of strains that are very low in terms of prevalence in meat, but pretty far to the right on the x-axis, which could mean, maybe our model's broken, or it's just inherent. And some strains are more inherently virulent, and those were the groups, the phylogroups that we know are inherently more associated with XPEC. And so it all kind of actually made sense, but I didn't explain it to where it would make sense to a general audience. I apologize.
That might well be me. So
Yeah.
thank you.
Thank you for asking me to clarify. Yeah.
Hi, great talk, really interesting.
Thank
I
you.
was wondering, in your human samples that you've collected, if you have any information on, or you any thoughts really on like if dietary preferences causing any differences, or like if you have a bunch of vegetarians, are they getting less of these infections?
Yeah, so I have a lot of thoughts on that. Maybe I do speculate a bit. But Amy Mangus, a Canadian who now, formerly at McGill, now in BC, she did some studies, some epidemiologic work. She was a real pioneer in this area, by the way. I'm following in her footsteps. But she did some proper epi studies where she looked at poultry consumption, and women who had more poultry consumption exposure had higher risk of getting UTIs. And so I think there are some studies suggesting that. What was the second part? Sorry.
That was it.
Yeah, for our study, I mean, because we're trying to work at a scale, we're just getting we're collecting the isolates and we're not getting any patient information. Yeah.
We have time for one more question.
Butterfly.
Yes. Hello? Hello?
Hello.
Yeah. Mohammed Al-Fatah, Animal Science Department at McGill University. So I'm curious about the meat samples. Like, do you have any idea like where this E. coli acquired through the production process? Like, where is that at the farm level or at the slaughterhouse level, or which level of production this E. coli strains acquired?
Yeah, so we have a lot more meat samples than we have animal samples, but there's some pretty good consistency between what's in the animals and what's on the meat. And you see different strains associated with different meat types. And so I think it's mostly coming from the animals themselves. I mean, you could have some strains that are established in the slaughter facilities, but I think ultimately they're coming from the animals.
So I'm the co-chair of the session. I'm Bing Guo from University of Surrey in the UK. It's my pleasure to invite our next speaker, Anna Barakarasiolo, to present constructed wetland and antimicrobials.
Thank you very much, Chair, for the presentation. I will present to you some results on real case studies of coastal wetlands. As is known, wastewater treatment plants are not effective in removing emerging contaminants. And at the same time, we know that constructed wetlands or treatment wetlands are the most well-known nature-based solutions used to treat wastewater. In this case, plants support microbial communities in different ways, and plants make it possible the coexistence of microaerobic and anaerobic environment in the same place, where the relationships between microorganisms and plants can be so strict, so close, that we consider the system of a plant and its microbiome as a meta-organism. Consequently, this system, this meta-organism can have a great potential to not only degrade organic matter, but also to transform and detoxify organic contaminants. However, until now, knowledge on the effectiveness of constructed wetland in real case studies are scarce. Most of studies in literatures came from mesocom studies, but few examples are on real treatment wetlands. In this work, we want to show this work deals with evaluating not only the capability of a real wetland to degrade organic matter, but also to investigate if the system is able to decrease emerging contaminant, considering in this class also antibiotics and antibiotic resistant genes. This was possible using an ecological approach. An ecological approach wants to combine physicochemical and microbiological analysis. Sampling were performed of water in different points at the inlet, middle, and outlet point, and at the same time, sediment from the vertical and rate of flow were also performed in correspondence of the Phragmites australis rhizosphere. Chemical analyses were performed for 1 year every 3 weeks between October 2022 and July 2023. Microbiological analyses were performed once for each season, once in autumn, once in winter, once in spring, and once in summer. The constructed wetlands is located in the countryside, Danish countryside, and it is a small constructed wetlands. It receives wastewater from a small village of about 100 people. It has been working since 2014. The inlet point where the wastewater is collected, receive at the same time rainwater and can collect water from the surrounding agriculture areas. You can see in this picture how the system works. There is a collection the water goes in the vertical flue beds, that is anaerobic flue beds, then through a pipe in the middle point, it goes to the rate of flow, and from the rate of flow to the outlet. So during this talk, we will report results about the inlet, middle, and the outlet points that deals with water, and about the vertical flow and the area of flow that are sediment samples results. Chemical analyses were performed in situ for temperature, pH, electroconductivity, and organic carbon, nitrogen, and the 71 macrocontaminants, including household products, pharmaceutical, biocide, and antibiotics, were analyzed. At the same time, microbial analyses were performed. In this slide, you can see that the system was able to decrease efficiently the organic load in terms of organic carbon and nitrogen. And the interesting point is that the temperature of the water maintained between 4 and 22 degrees. This shows that even if the air temperature can be much lower, this is a sort of environment where bacteria can be protected. In this slide, you can see, as an average of the different samplings, the overall results of all emerging contaminants, and you can see in blue the inlet water, in red the middle point, and in green the outlet point. You can see that only a small number of the emerging contaminants searched were found, and with different load between autumn, winter, spring, and summer. Highest contaminant load was found in summer. But this can be explained by the fact that the treatment system receive not only wastewater but also rainwater. So in the other seasons, except in summer, we can observe a dilution effect of the water or the rainwater. In this table, we can see an overview of the sum of all the overall emerging contaminants. You can see that the total amount in the inlet, at the middle point, at the outlet. In any case, the system was able to remove the emerging contaminants with a percentage varying from 58% in winter to 80% in summer, considering that summer was the period in which we have the highest load. Of just 16 compounds were found. Most of them were pharmaceuticals, and antibiotics were only sporadically found. This is an important point because in the table on the right, in this table, you can see underlined the sampling time for antibiotic resistant genes analysis. And in most cases, in correspondence of the sampling, we didn't find any antibiotic. So we can say that antibiotics were not ubiquitous common contaminants in this system. In this slide, you can see the microbial abundance that was calculated by direct counts method using a perfluorescence microscope. And it's very interesting to note that there is a high load of bacteria from the inlet, but the system is able to decrease significantly in the number from until amount that are very similar to that found in surface water. At the same time, the sediment was able to decrease the number of microorganisms, and this means that many bacteria which entered in the system from the wastewater, including antibiotic-resistant bacteria and pathogen bacteria, decreased. Regarding antibiotic-resistant genes, we found antibiotic-resistant genes versus sulfonamides. And as you can see, considering the inlet, middle point, and the outlets, we found mainly SUL1, SUL2, and INT1. And in each season, their relative abundance decreased significantly from the inlet to the outlet point. And this means that the system was able to decrease significantly antibiotic-resistant genes number. Anyway, in winter, higher abundance at the middle point of disulfonamide genes was observed. In a similar way, if we look at the results of the sediment samples, we find that the effectiveness of the system to decrease from the vertical flow to the lateral flow the antibiotic-resistant genes, but this phenomenon was less evident in winter. So there is a sort of accumulation of antibiotic-resistant genes in the winter seasons. Regarding microbial community, you can see that in the sediment corresponding to the vertical flow, the lateral flow, the biodiversity was much higher than the water. And this was possible because in this case we had resident bacteria that lived in relation with the roots that were presumably able to outcompete allochthonous species entering in the system through the water. Water microbial community, sediment microbial community was also analyzed, and in general we can tell that we identified bacteria genera that were able to remove pharmaceuticals and antibiotics. Moreover, also bacteria involved in nitrogen and phosphorus cycle were found. Most of bacterial genera associated to pathogen species also decrease significantly from the inlet to the outlet. In conclusion, the vertical system was very effective, but in general, the treatment wetland was effective in decreasing both the emerging contaminants, overall emerging contaminants, and antibiotic-resistant genes. And this was presumably possible to the synergic interaction of the Phragmites australis and its rhizosphere microbiome. It's very interesting to note that in the winter, where the plant is in its rest vegetative, its effectiveness was much lower, and probably temperature was very important not only for the plant, but also for the microorganism activity. We cannot exclude that in winter some antibiotic-resistant bacteria can maintain their resistant genes as a response of stress, considering that the temperature, the lower temperature never was lower than 4 degrees. That is a low temperature for some bacteria, but not so low to completely outcompete some species. These results are among the first data on emerging contaminants RG removal in the case of a real treatment wetland. Of course, this is an example of a small plant for few people, and it would be very nice to look at higher treatment wetlands that receive water from more people than 100 people. Thank you.
Jan Siemens from Gießen, Germany. Thanks for this very interesting talk on a very interesting alternative for smaller communities. Regarding the removal of your contaminants, I was wondering whether you have a build-up of concentrations of these contaminants in your sediment, so that the sediment just act as a filter, accumulating these contaminants over time, like metals, for example. And the question then arises, of course, that whether over time you build a kind of bioreactor that is a perfect environment for selecting resistant bacteria in the long run. So what is your thought about that?
Okay, thank you for your questions. Of course, it would be nice to analyze the chemicals inside the sediment and also for a longer time. We don't not exclude that we can do in the future, but these are the first results of one year survey, and we were a little bit surprised for these results because we didn't know what could happen, but your suggestion is very good. We can do in the coming future.
Can I ask a question? I really like the study into constructed wetland because it's getting more and more constructed in Europe and in the UK. But from an engineering perspective, you have different hydraulic retention time. You have different design factors. Have you considered these factors into the results?
Okay, really, I am not an engineer, I am not expert of this aspect because this constructed wetland was built in Danish from a company that design normally this kind of constructed wetlands for in commercial market. So I think that they are sufficient expert in reply to your questions, but really I am a microbiologist and I am not able really to go so in detail for answer. But I think that the technology is in advanced phase, so I think that it's possible to adapt to different situations. Yes.
Question. Juliana Calabria, Minas Gerais, Brazil. I was wondering about the hydraulic detention time of the vertical flow wetland and also of the horizontal flow. Could you tell us about this?
Well, the retention time was few days. And I don't understand exactly your question.
Because if I understood correct, you have two wetlands in series. Is that right?
Two beds.
Flow.
The wetlands is one. There are two beds that one close to the other. Yes, one is anaerobic and the second one is aerated.
So when you mean 2 days of hydraulic detention time, you mean in total?
In total.
Okay. Okay.
Okay, the detention time is for overall the system.
Okay. Thank you.
Good morning, everyone. I am Kiran Mee. I am a PhD student at Auburn University, United States. Today, I will talk with you about our pilot study on environmental antimicrobial resistance threats to food safety and security. We know the overusage of antibiotics in food-producing animals led to the emergence of antibiotic-resistant organisms and residues. So they shed antibiotic-resistant organisms and residues. So when their manure is used as a fertilizer, it can contaminate the environment with these pollutants. So these pollutants, they can affect the food safety if they get transferred to the food farm produce, ultimately reaching the human beings. What is still unknown is if using this manure as a fertilizer can affect the food security. That is, for example, the usage of poultry litter as a fertilizer, it is an economically viable way to recycle the resources and increase the crop production. But polluting the environment with these pollutants, antibiotic resistance, can affect the pollinators' health, causing dysbiosis and finally predisposing them to the diseases. Why we are concerned about pollinators? Why this is more important? As 1 out of every 3 bites of food we eat is pollinated by bees. All these fruits, vegetables, and field crops, all those that are pollinated by the bees, they will be off the menu if there are no bees. So understanding the impact of agriculture practices on the pollinators' health is very essential for food security. So to determine the effect of using poultry litter as a fertilizer on the pollinators' health, we conducted our experiments in 2 high tunnels. And these tunnels were divided into 3 equal blocks. And we fertilized the soil either with fresh poultry litter or composted. As composted is known to reduce the load of pathogenic organisms, we wanted to compare the use of fresh poultry litter and composted on the pollinators' health. And we chose squash as our crop of interest as it is highly pollinator dependent and is attracted to bees. And before introducing the bees, we made sure that those blocks are divided by the nets so that there is no movement of bees from one block to the other. Our objectives and hypotheses are, our first objective is to see the change in bees microbiome after being exposed to the environment fertilized with poultry litter. And we hypothesize that there will be change in microbiome and increase of bad bacteria and decrease of good bacteria, predisposing the bees to pathogens. So we will also measure the prevalence of pathogens after being exposed to the environment fertilized with poultry litter. Our second objective is to see the AMR change in bees over time. And we hypothesize that there will be increase of AMR in bees after being exposed to the polluted environment. We will also compare the AMR change in bees that were exposed to the environment with fresh poultry litter and composted. And we hypothesize that bees that were exposed to the environment fertilized with fresh poultry litter will carry more AMR than the bees that are exposed to the compost, as composting is known to reduce the load of pathogens. Coming to the timeline of events that we performed in our experiments, on day -63, first we collected the baseline soil samples from the tunnels to see the initial AMR levels. And on the same day, we fertilized the soils with either fresh poultry litter or composted. 3 days after fertilization, we collected the soil samples to see the AMR levels after fertilization, and we planted the squash seeds. And on day 0, that is 2 months after the squash seeds have flowered, we introduced the honeybees. We had total 5 sample collection times, and these are sample types. Before talking about the sample types, I would like to tell you briefly about the bees. We call the group of bees as colony. In the colony, there will be eggs, larvae, pupa, and the bees, foraging bees and nursing bees. The foraging bees they are adult bees, but they can't fly. They have emerged from pupae. So they just take care of the bees, that is larva, the brood. And whereas the foraging bees, they go out and bring pollen, food, all those water sources that are essential for this colony. And then we collected all these baseline samples on day 0. They are soil, pollen, foraging bees, nursing bees, and larvae. And we collected same sample types on the subsequent sample collection times. We processed all these samples for the data, but for shotgun metagenomic sequencing, we focused on subset of samples from day 0, day 21, and day 56. And we isolated DNA from all the samples, fertilizers, soil, bees, and larvae. And we sent the samples for metagenomic shotgun sequencing. And microbiome and resistom analysis was performed by our postdoc, Dr. Alini. For culture-based approach, we targeted highly pathogenic bacteria of bees, that is Pentibacillus larvae, that causes American foul brood disease, and Melissococcus plutenaris, that causes European foul brood disease. We also targeted the opportunistic pathogens, that is Serratia and ecoli, and also the commensal bacteria, that is the good bacteria, Gilliamella apicola. Coming to the results, the first objective was to see the change in microbiome. For microbiome data, we focused on soil, foraging bees, and nursing bees. First, we wanted to compare the overall microbiome composition of these 3 samples, and we could see that the soil microbiome is markedly different and has different microbiome composition when compared to bees. But the bees microbiome is very simple, and it is being dominated by 3 phyla. They are Actinobacteria, Firmicutes, and Proteobacteria. However, there were no shared bacteria observed between the soil and bees, showing that the microorganisms did not colonize the highly specific bee microbiome. Next, we wanted to see closely compare the microbiome composition of foraging bees and nursing bees exposed to litter environment fertilized with fresh poultry litter and then compost over time. Here we could see, though they shared majority of the microorganisms, still we could see there is differences in their abundances. And interestingly, Bartonella, that is the common bacteria in bees, but is not the part of core bacteria, was prevalent more in the foraging bees that were exposed to the litter than the compost. And the same trend was almost observed in the nursing bees too. And overall, we found that the microbiome of bees, of foraging bees, exposed to the compost and litter is significantly different. The fertilizations, it significantly impacted the microbiome of these foraging bees. Our next question is if this dysbiosis is sufficient to increase the prevalence of pathogens. So, and through our culture-based methods, we found that the very highly pathogenic bacteria of bees, that is Penibacillus larvae and Mesococcus plutinus, they were never detected in our bees in these experiments. However, the opportunistic pathogens, Serratia and E. coli, they started increasing on day 21, which coincided with a decrease of G. apicola, the good bacteria, on day 21. This also coincided with our metagenomic data, where we observed the increase of opportunistic pathogens on day 21. All this together might indicate that the dysbiosis event might have taken place on 21, exposing the bees to more to opportunistic pathogens and the detection after day 21. So next, our objective was to see the AMR change in bees over time and also comparison between the litter and the compost. As I said, that we wanted to see the impact of using fresh poultry litter and composted. First, we measured the relative frequency of antibiotic resistance genes and mobile genetic elements using qPCR, and we found that the litter had, as hypothesis, litter had more AMR levels than composted, except for the class of sulfonamides. Our metagenomic data showed that the samples, the soil, and the bees that were exposed to litter had higher args richness than the bees that were exposed to the compost. And we also observed the peak on day 21 in both the bees, that is nursing bees and foraging bees, which matched with the dysbiosis event on day 21. Though this was not in correlation with the microbiome richness, so there would be chance that this args increased not because of the new bacteria from the bees, but might be due to the mobile genetic elements. Next, we wanted to explore the resistome composition of soil and the bees. Here we could see that the red and blue bars, that tetracycline and MLSB resistant genes were dominating the resistome of bees. They happen to be the most commonly used antibiotic in both bee settings and also the poultry. Here we could see that soil resistome it has a very different profile, and it's with much higher diversity. Though there are differences in the resistome, we wanted to see if there are any shared arcs between these two environments, that is soil and the bees. And we found that 34 genes are being shared between these two environments, most being the tetracycline, polymyxin, and beta-lactam, and also MLSP. So these happen to be the most commonly found antibiotic resistance genes in plasmids too. However, we also found that the macrolide resistance gene, that is MACB, it was more, it shared a lot of genotypic diversity between both the samples, that is soil and bees, that suggesting that there could be no transit of arcs between them. So overall together, we could say that there could be transit of arcs between the soil and the bees, maybe not with the new organisms, not sharing the organisms with the soil, but might be due to the mobile genetic elements. So with the data we have, I would like to conclude that we observed that there was a disruption of microbiome observed after being exposed to the environment fertilized with poultry litter. And also we also observed that AMR diversity in the bees. And also we observed that fresh poultry litter had more impact on the both bees microbiome and resistome, suggesting that these agriculture practices might have impact on the pollinator cells. So our future studies will focus on understanding the mechanism of increase of AMR in bees exposed to the fertilizer, if there is transmission from the fertilizer to soil, soil to pollen, pollen to bees, by reconstituting the genomes, or if the increase in AMR is just due to the antibiotic pressure by measuring the residues. With this, I would like to thank our team and also the people who are involved here, the agriculture people and B-Lab people. And I still remember the day the germination of all the seeds and the flowers, and I knew the pain of the agriculture farmers, how painful it is if the plants don't show up. It was really exciting to work with the bees and stuff, and I would really like to thank our funding. And you could see the QR code of our lab. Our projects are all on AMR. You could scan to look into our works. And thank you so much for listening to us.
Hi, lovely talk, thank you. I'm a beekeeper, so that was really interesting. Sorry if I missed it. Did you have a control set of bees that you raised
Actually,
in the
we
lab
expected that compost will have less, so we couldn't have controls. The squash, it wouldn't go without any fertilizer, so we couldn't have. And the future studies are like to work on the rotational crops, so we don't have to use any fertilizer. So then we will have control. Initially, we expected that compost will have very less and fresh will have more, so we couldn't.
Sure. One last question. The hive itself, was it brand new wax that you put in the frames in the beehive?
Sorry?
The hive that you were keeping the honeybees in, was it a brand new hive with fresh wax? It hadn't previously been lived in?
Actually, we got them from the bee lab where they maintain those colonies. They were from the same, actually they are from the same area, so we could consider them as baseline from the same area.
Cool. Awesome. Thank you.
Thank you.
There's a question there.
Ziggy Kulik from the Public Health Agency of Canada. Lovely talk, thank you. Just interested, you focused on domestic honeybees. Did you consider using native ones?
Native? Sorry?
Native bees that pollinate squash?
No, we use Apis mellifera, the bees.
Okay, thank you.